Hi everyone and welcome to our GCSE Biology lesson. In today's lesson we are going to be looking at the digestive system, and in specific we are going to be focusing on enzymes. Okay, so if we make a start now. So as I've mentioned, we are going to be focusing on the first sort of part of enzymes. So before we move on to enzymes, we're going to have a look at cell organization, and that's what this whole unit is based upon. So before we move into this subtopic of enzymes, we need to focus on what this unit is about, and that is cell organization. So cells are basic building blocks of all living organisms. So every small part of an organism starts off from cells. These cells then build up together to form the human organisms. Okay, so what you need to know here is that cells are the basic building blocks of all living organisms. Now, a tissue is a group of cells with similar structure and function. Organs are aggregations of tissues performing specific functions. So organs are a group of tissues, and then organs are then put into what you call an organ system, which work together to form an organism. So what you basically need to know is that we've got a cell as the simplest building block, a group of cells performing a similar function makes a tissue, a group of tissues forming a similar function groups into an organ, a group of organs performing a similar function is then grouped into an organ system, and then the organ systems then group together to form an organism. So in today's lesson, we are going to be focusing on enzymes, which are part of the digestive system. So that's an example of an organ system. You've got the nervous system, you've got the circulatory system, you've got the immune system. Those are all examples of organ systems. Now, an example of an organ may be the stomach. Okay, so we know that the stomach has an important role in the digestive system, along with you've got the other, like the small intestine, the large intestine, all of the organs, they work together to form the digestive system. And then you've got cells, you've got tissue, and the organism is basically the human body. The whole human body is the organism that we are talking about in this case. So what you need to know from this slide is that the cells are the basic building blocks of life. Then you've come across a tissue with a group of cells forming a similar structure and function, and then organs are then aggregations of tissues forming similar functions, and then organs are organized into organ systems which work together to form an organ system. So basically you need to know this flow diagram that I've drawn here. So if we move on now to look at enzymes. Now there are three key points you need to know about enzymes. Now enzymes are biological catalysts. So these are the 3 points you need to know about enzymes. And then we're going to move on to the lock and key theory. So enzymes are biological catalysts. That's the key phrase there. Enzymes are biological catalysts. Now, if we break this phrase apart, bio is about the body. Logical is about function. And now catalysts, they speed up a reaction. Okay, so you need to know what a catalyst is, and a catalyst is something which speeds up a reaction. So a catalyst, we say, catalyzes a reaction. So it speeds up a reaction without being used up in the process. That's what a catalyst is. So enzymes are biological catalysts. So they're body functioning catalysts. So they are catalysts which allow the body to function. So enzymes are catalysts which allow the body to function. That's what you need to know there. They speed up a reaction without being used up in the process. And they speed up the reaction by providing an alternative pathway. So if I show you an example here, let's say we have our original sort of store over here, and then we have our end here. Let's say that this sort of route is sort of full. Okay, there's so much traffic down this road. The enzyme will form an alternative pathway to get to the end line, and that's how it catalyzes the reaction. If too many chemical reactions are occurring in one direction, then it will find an alternative pathway to get to the finish line. And that's how enzymes work. The key thing, the key phrases you need to know to get the marks include: you need to know biological catalysts, speed up a reaction without being used up in the process, and they provide an alternative pathway. So in an exam, if you're given this question, it would normally be worth 3 marks. Okay, so now if we move on to the lock and key theory, now this is a really common exam question, so you need to know the key points along with the lock and key theory. Now the lock and key theory is basically as it sounds. You've got the lock and you've got the key. Now you know that the lock, you know what a lock is, and you know that key is used to open the lock. So key is used to open the lock. And that's basically what happens in terms of enzymes. In this case, you've got the key as the enzyme, and then you've got the lock as the substrate. Okay, so we've got the, let's say we've got sort of we've got an enzyme here. This is our enzyme. And then we've got our substrate here. I'll go through these words. Now, the substrate is basically, let's say we've eaten food. I'm just going to give you an example here. Let's say we've just eaten food. The food molecules are going to be large. Okay, they're going to be large. How are we going to break these down? So let's say each food molecule is a substrate. Okay, each food molecule is a substrate. Now this substrate is going to go into the enzyme. The enzyme is going to break it down. Okay, the enzyme is an example of a protein. Now enzymes are going to break down the substrate. So let's say the food that we've just eaten, so this is our food that we've just eaten, is the substrate. We want to break it down as small as we can so we can digest it. So enzymes, remember the key parts we looked at, they are biological catalysts, speed up a reaction by providing an alternative pathway. Let's say in this case that this is a chemical reaction. This has to be broken down so a chemical reaction can occur. The enzyme, whatever is being broken down, is known as the substrate. The substrate is whatever needs to be broken down. So the substrate is whatever needs to be broken down. Each enzyme has a specific active site, and the active site is just basically this part. The part where the substrate is going to fit onto is called the active site. Okay, so this is what you call the active site of the enzyme. And the active site is basically the part where the substrate is going to attach itself onto. So where the substrate is going to attach itself onto. So if we have a look at this case, so whatever is being broken down is known as the substrate. So we've got the substrate here. We've got the substrate there. We've got the enzyme here. So we've got the enzyme. And then each enzyme has a specific active site. That's the active site. So just remember that each enzyme has a different specific active site. Each enzyme has a different and specific active site. So let's say that this enzyme is specific to this substrate. That means that only this can fit into this enzyme. No other. So if you had something like this is not going to be able to fit inside that enzyme. Only this substrate will fit into this enzyme. Now, once it's fitted in to the enzyme, so let's say that the substrate has fitted in, the enzyme will break the substrate down into its products. Okay, so the enzyme will break substrate down. And then it will form its sort of products. Let's say in this case, it forms two smaller products. So the enzyme has catalyzed the reaction. Has catalyzed the reaction. So in this case, what we're looking at is that the enzyme is breaking down the substrate into its products. If I go through the key parts, so we've got the enzyme, which is a protein. Each enzyme has a specific active site. Remember that it has a specific active site. The substrate, it's whatever needs to be broken down. The substrate binds into the active site of the enzyme. Okay, you need to use this terminology. The substrate binds itself onto the enzyme's active site. Okay, and remember, each enzyme has a different active site, which is going to be specific to a substrate. So this substrate is going to bind onto this enzyme's active site. And then the enzyme will break the substrate down into its smaller products. Okay, as you can see there. So that basically goes over the lock and key theory. If I go through the keywords over here, you need to know about the active site, the substrate, catalyze, specific, and binding site. So basically, if I explain it now, we've got the enzyme. You need to remember that each enzyme has a specific active site. And this is the layer over here, what we find. This is this layer over here, where the substrate is going to bind itself onto. That's called the active site. Okay, and the active site is where the substrate is going to bind onto. And we've included the binding site here as well. Okay, the active site is also known as the binding site, where the substrate is going to bind onto. Now the substrate is where the substrate is what's going to be broken down. Substrate is what is actually going to be broken down. Okay, catalyze means to speed up the reaction. So the enzyme is a biological catalyst. So the enzyme catalyzes a reaction. So an enzyme catalyzes a chemical reaction by providing an alternative pathway. And remember, each enzyme is specific to its substrate. So each enzyme is specific to its substrate. Now, what you need to know here is that an enzyme shaped like this will only bind to its matching substrate. If we had an enzyme such as this one, this will not fit in this enzyme. Okay, because each enzyme is specific to its substrate. So each enzyme has a different active site, so it can bind to its specific substrate. Okay, so the key things again, I'm going to highlight, has a specific active site. The active site is where the substrate is going to bind onto. The substrate is what's actually going to be broken down. An enzyme catalyzes a chemical reaction, and each enzyme is specific to its substrate. Okay, and whenever you're given a question to do with enzymes, what you need to remember is you need to include these words, and normally the marks come from including these words. So if we look at what we've looked at in today's lesson, we have looked at cell organization. We saw the building blocks of the human body. So we start off from a normal cell. A group of cells performing a similar function then gets characterized into a tissue, and then a group of tissues forming a similar function. Sorry about that. Okay, so a group of tissues forming a similar function forms an organ, and then a group of organs forming a similar function forms an organ system. And then a group of organ systems such as the nervous system, the digestive system, you've got loads of organ systems, you should probably be able to name some as well. A group of organ systems then form an organism. Okay, so we then looked at enzymes. We saw that enzymes are biological catalysts. And we saw where the 3 marks are coming from. Biological catalyst, speed up a reaction, by providing an alternative pathway without being used up in the process. And we saw how that actually happens. Again, common exam question, normally at the start of your exam paper, and normally people lose either 1 or 2 marks. So it's really important you remember where the 3 marks are actually coming from. Okay, moving on. If we look at the lock and key theory, which we did look at, I'm just going to skip to this slide as this explains it a bit better. So we saw that the substrate is what's being broken down. Enzyme, we know that it catalyzes a reaction. We know that the substrate binds to the enzyme's active site. The active site is just this layer which we saw in this case, this layer over here. And each substrate is specific to the enzyme's active site. And each enzyme has a different active site because each enzyme catalyzes a specific reaction. Okay, and then the substrate we saw binds to the enzyme's active site, and then the enzyme breaks it into its products. Okay, and we saw this is a keyword again, specific. I need to emphasize this word specific. It's really key and many people forget to state that word. Okay, so we've looked at everything for today's lesson. Now, if you guys want to request a topic you would like to be taught in our lessons, then you can just fill out this form. It just asks for your name, email address, and the topic you would like to be taught. I'm going to paste it in the Q&A. Okay, so I've pasted the link to the form in a Q&A. I'm also pasting a link to buy me a coffee if you would like to thank us or support us for producing these free GCSE educational sessions. You can buy us a coffee using the link I've pasted in the Q&A. Again, it takes a lot of work and determination and preparation to set up these lessons to be taught for free. And we're doing it weekly, so we would really appreciate it if you would be able to buy us a coffee. Now, the link to the form I've posted in the Q&A. If you would like any specific topic to be taught in our free GCSE sessions, then again, just fill out the form. Okay, so you should come across something like this. So if you say start now, it just asks you 3 simple questions. Your full name, email address, and what topic would you like us to cover in our live lesson. So if you would like any topics you specifically struggle with, let us know and we would be more than happy to cover them for you. We will send you an email of which lesson they will be covered in, and then you would be able to join that lesson. And the Buy Me a Coffee link is in the Q&A if you would like to thank us or support us for producing these free GCSE educational sessions. If you have any questions, you can let me know now in the Q&A. I'll stay on for a couple of minutes. But if you don't have any questions, then you can leave the meeting.